All right, welcome everyone. We're gonna be starting. So for people online, if you have any questions, please use the Q&A tab. And if you have technical issues, let us know in the chat. All right, so it's a pleasure to introduce David on behalf of Nacho Bravo, David's host, who cannot be with us today. So he just shared a few words to introduce David. So welcome, David. All right, so David is a researcher at MIVEJEC, where he works on behavioral, sensory, and chemical ecology. David completed his PhD at the Swedish University of Agricultural Sciences. No, it's a mistake, sorry. So we will complain to Nacho. But it was in Sweden, right?
No, it was in Finland.
In Finland.
Same thing. Same thing.
Sorry.
I didn't say that.
Focusing on food and oviposition preference in Drosophila flies, is that correct?
No, not at all.
Okay. Should I continue?
I don't know.
Actually, it's quite funny, so let's go on. He then carried out several postdoctoral positions in Sweden.
Yes.
Yes.
Yeah.
Where he developed strong expertise in insect neurophysiology and chemical ecology.
Yeah.
And then today at MIVEJEC, David works on disease-transmitting mosquitoes.
Yeah.
Yep. That's pictures here. And the behavioral mechanisms underlying resistance to insecticides and repellents. And by combining neurophysiology, behavioral ecology, and chemical analysis, it contributes to the development of innovative and more targeted strategies for vector control.
Yeah.
So please welcome, David.
Thank you very much. Huge thanks to Nacho for this nice introduction. So I'm going to talk what I really like is to look at the insect brains and especially their behavior. So I will start by disagreeing with you most probably while I say that mosquitoes are wonderful insects. So most of you might say like, no, it's not true. Well, I think they are because they express different, you can find them almost everywhere. They express different ecological and behavioral patterns, which are extremely nice. And there are about like 3,500 species of mosquitoes, but only 5% bite or bother people. Problem is that this 5%, they are extremely nice at the, I don't know whether, can we remove this or not? Well, anyways, so they are very good at hosting and transmitting viruses and parasites. So once mosquito bites an infected host, then it will transmit it very readily. As here. Yeah. Thank you very much. Yeah. So they are very good at transmitting viruses and parasites, like for instance the malaria parasites, Plasmodium, which are associated to a genus of mosquitoes called Anopheles. Another, the arboviruses from dengue, Zika, chikungunya, yellow fever, which are associated to Aedes, and for instance, West Nile viruses, which are associated to the genus Culex species, among many others. So this makes them a public health problem, where half of the world's population is at risk. Thing is that because of several species that are very invasive, that are like very good vectors as well, like Aedes albopictus, which is the tiger mosquito that you might well know him, and Anopheles stephensi now spreading in the whole Africa, which is a very good vector of malaria. These half world's population is at risk, is increasing. So we should be aware of that. So to protect us against these mosquitoes, we have developed very many different vector control and transmission prevention tools, like, for instance, the new techniques as the incompatible insect technique with Wolbachia, which here you're expert at ISM, the genetic control as the sterile insect technique, but also more common things or more, we've been for hundreds of years, like repellents. Also like olfactory-based traps with VOCs, with volatile organic compounds to attract them and get them, or also the chemical to control with insecticides, which is the most used. So all these traps and strategies and techniques are based on the species ecology and behavior. So you will use one depending on the behavior of the species. So I will focus in this talk in this chemical control insecticides because it has been used on very effectively, proven be very good in controlling certain mosquito species. So, and I will focus in what the mosquito I'm studying is. An Anopheles mosquito is transmitting malaria. We found it in sub-Saharan Africa. It's very well spread. And malaria disease, in the last WHO report in 2025, in 2024, there were like 282 million cases, estimated cases, which 610,000 deaths, of which 75% are children under 5 years old. So basically, mosquito might infect these hosts and then transmit the disease very, very readily. So we have been using insecticides for a very long time to try to control this malaria. And how do we do it? We do it mainly with two techniques. One is insecticide-treated nets that we put around beds, so people is protected while they are sleeping, and also using another technique, spraying insecticides to the walls and so on, so the mosquitoes, when they go and rest, they get into contact with insecticides and they die. Okay, so these techniques are based on the behavioral traits, because we know that Anopheles mosquitoes, they bite people, they prefer to bite people, they do it during the night, and they do it indoors. So they work very well. So thanks to these techniques, from the beginning of the century in the year 2000, we can see millennials. So we started huge programs for distributing and treating houses with insecticides. Malaria, we saw a very decrease, a nice decrease of malaria incidence in the populations, and 80% of this reduction has been attributed to the use of insecticides, like insecticide-treated nets on indoor residual spray. But from the 2015 onwards, we see that this decline, this reduction of malaria cases has stalled, and in some countries has even decreased. And we think, and we kind of like, we are very confident to say that this is because of the development of insecticide resistance. Okay, insecticide resistance has been classified in kind of like three major groups. Like we can find morphological resistance, which for instance is like cuticle thickness. It increases the cuticle thickness, the insecticide cannot really penetrate or less penetrate to the mosquito, so it doesn't die. But also, and the most by far studied mechanisms are physiological and molecular mechanisms, where, for instance, the target mutations that in the protein where the insecticide needs to act, there is a certain modification and the insecticide doesn't fit very well, so the mosquitoes become resistant. And also because of there is an increased activity of metabolic enzymes when the insecticide penetrates the mosquitoes, there is enzymes will cut the insecticide. The enzymes will cut the insecticide and then they will degrade it and it won't take action. But then more and more, there is more and more evidence that there is also behavioral resistance. And these we try to, so in the case of morphological and physiological and molecular resistance, it's just a selection process. You have a circular population where you have a part of this population which are, they have some certain modifications. And it is supposed that the most contributing is standard genetic variation. So these mutations are in the population. But there can be also in less extent de novo mutations or migration of certain genes, resistant alleles, into the population. So, and these stressors have been identified as for instance, the misuse of insecticides, like horrible use of this, usually as well, and this has contributed very well to this resistance, is the use of the same insecticides in agriculture. But also there are more evidence saying like it's a response to stress pollutants that are present in waters like heavy metals, but also a little bit to vector control. So these are the selective pressures that could act for these selection processes. But then with Thierry here and other colleagues at Mivesec, we thought that we should contribute or try to classify what we expect that it could be, or that could be that these behavioral modifications that they could contribute to the behavioral resistance. Well, that was not very clear, but so anyways, so imagine that you have some mosquitoes that however is that? So yeah, so we try to classify this behavioral resistance on different modifications that we could find, and they are hypothetical. Some of them we have proofs from the field. So the first kind of like classification, we could say like there are qualitative modifications, for instance, temporal avoidance, spatial avoidance, or trophic avoidance. These qualitative modifications prevent mosquitoes to get into contact with insecticides. So basically, they don't get into contact. They just like do all things, and they will never get into contact. So mosquitoes will be there, but they won't die. That would be kind of like an analog of enemy-free space for the people that can work with host or everywhere around interactions, something like that, more parasitoids. Then there is another classification, which is a quantitative modification or behavioral modifications, which could be that these would produce the amount of insecticide that mosquito take into their body. For instance, escaping mosquito when touches the insecticide, if it's fast enough, escapes fast enough, then it would be less exposed to this insecticide, less molecules will enter to the mosquito, so it will also survive. But also there are more hypothetical things like behavioral fever, behavioral thermoregulation that could activate physiological patterns to degrade these insecticides. But also, and Thierry is expert in this, is prophylactic curative medication. Could mosquitoes feed on something, some nectars, antioxidants, or whatever that could help to degrade or cope with this or reduce the amount of insecticides in their bodies. Then there is another classification that would be like modifications that would have on tolerance. For instance, here the mosquito gets the insecticide, but will do the best to compensate this presence of insecticide by, for instance, frequent compensation. If he got a sublethal dose, so he's going to die soon, or he's feeling bad, and so on, can just lay all the eggs and try to maximize the fitness that he can do. Also, tolerance thermoregulation, same thing, and tolerance medication. Here, doesn't reduce the amount of insecticide, but try to do their best to continue living and try to maximize the fitness. And then the last case would be like, since we know that there are plenty of insecticide resistant, like physiological, molecular, morphological, the mosquitoes with this resistance, could mosquitoes use this information in a way, in an associative learning way, saying like, okay, I'm resistant, I contact the insecticide, but I don't die. I still get my blood meal, and then later on I will use these insecticides as a proxy of the presence of a human being. So these are, most of them are hypothetical. Escaping has been proven with BDT and Drosophila, which could be selected. And this is what I will concentrate, because it's the ones that we have more and more evidence in the field. So I told you that Anopheles mosquitoes prefer like people at night and indoors. So when we put the insecticides, we see nowadays that some of them prefer to feed on animals, which would be excellent because for the parasite is a dead end. But some of them, or most of them, they have changed their rhythms, and they bite before or after the night. So what happens here is that people are not protected during this time, so they get bitten while they are, for instance, outside. And we see as well that this indoor preference for biting, we can see it now that it's moving towards outdoors, and there people are not protected. So given genetic variability present in the population, if the mosquitoes are sensitive to insecticides and try and get into contact with insecticides while trying to get a blood meal, they will die. Okay, great. But if they are resistant, they will feed and nothing will happen, and they will continue lay their eggs and so on. So in a matter of few generations, I expect the resistant phenotypes will be measured in the population. And taking into account that this larval, there is a larval selection on the breeding sites, but also an adult selection with the same compounds. And this selection process will be very, very, very fast. Okay, but there might be other variants already in the population that they do not prefer to bite indoors, or perhaps they don't prefer to bite people, or they don't prefer to bite at night, but during the day or down at dusk. So these can be selected because they will never get into contact with insecticides. They will be there, but since there is strong selection pressure indoors, both people and during the night, they will be really present and really rapidly in the population. However, this temporal avoidance, spatial avoidance, trophic avoidance that could be achieved by just natural selection processes could be also achieved by phenotypic plasticity if and only if mosquitoes are able to detect insecticides beforehand. And this learning capacity as well. They need to sense what is going on and then to be able to associate and use this as a proxy. So this is what my project that was funded by the ANR is all about. Can mosquitoes detect insecticides? And you know that adaptive decision making to control resources and avoid stress sources requires specific sensory inputs. But can actually mosquitoes detect insecticides from a distance? So the most common used insecticides in vector control for Anopheles now is changing a little bit due to this resistance that we found are pyrethroids. For instance, cialotrin, deltametrin, cypermetrin, and permethrin, which are bio-inspired by pyrethrins that we find in what is it called? This Dalmatian pyrethrum, I think it's a chrysanthemum species on the pods of the seeds. So there you have a really nice insecticide, pyrethrins. They work very well, but they are not stable. So chemists, very clever chemists, they put some modified a little bit the molecule. They put some allergens here, and they made them more stable and very long lasting. So yeah, they can last very, even though they can be degraded, but they are much better than pyrethrins in this sense. So what we know from the field is that colleagues from IBESEC did a meta-analysis from different papers that way these papers, they were trying to see how these mosquito nets, different kinds of like brands with different insecticides combinations and so on, would affect the mosquitoes. So the experiments were easy. There were experimental huts where you put a mosquito net, and then experimental huts when you put control nets without insecticide, and you count the number of mosquitoes that get in, get out, and so on. And they found out that in many studies, they found that mosquitoes were not entering. And then because of certain procedures from the WHO to go to test for these nets and so on, they tell them to wash them several times to see how these long-lasting insecticide nets get old, and whether their efficacy is different or is just like stopped. And then they saw that some of them were like kind of like attractive. So this repellent effect or attractive effect both suggest that there is a spatial sensory detection that mosquitoes can detect these insecticides. But insecticides used, these pyrethroids used in nets and in residual indoor residual spraying, they have some problems for this hypothesis because chemically they are not expected to be volatile. They are very big molecules and with a very low vapor pressure. Physiologically, we don't know, or we didn't know, that mosquitoes would have chemoreceptors to detect these insecticides. And behaviorally, the evidence of this special repellency is debated, even though now these pyrethroids are being volatilized and they are used as special repellents. So somehow, yeah, there is something, but the ones used in nets and IRS are a bit different, less volatile, and they stick to everything. Okay. So this was part of the thesis, or most of the thesis of my old PhD student now, who was Sambis Kambu, who's now a doctor. So I will present the results that he got during his thesis. So the first thing, do mosquito nets emit? Can we catch these insecticides in a room where you put a mosquito net? So he did this in Burkina Faso. We wanted real conditions in the place where malaria is endemic. So he put a mosquito net in a laboratory, and then he started collecting the air, the indoor air that was in the room. And then we analyzed this, and we really see that when analyzed it with JCMS, we saw the permethrin in this case, but also we did it with deltamethrin and cypermethrin, and we found that we see that they are in the air. And after washing them and sun drying them, and here I will go a little bit more, because I mean, talking to some place, he was saying like, yeah, people don't use to, I mean, they don't wash the nets 20 times, and usually they wash it once, twice, or perhaps, yeah, no. But the most important is that they dry them in the sun. And then we know that these pyrethroids, when mixed with water and exposed to sun, they have double bonds that they can break. So there are potentially degradation compounds that could be emitted. And these degradation compounds are much more volatile than this. So we try to analyze this data, and for instance, we haven't finished analyzing this data for the what is it, degradation compounds, but I'm sure they are there, because we find them very, we're using all the techniques. But I can tell you that at least pyrethroids from mosquito nets are present in indoor air. Okay. The second one, we wanted to see, okay, now we will ask mosquitoes whether they can actually detect these insecticides. So for this, we wanted a proof of concept, can detect them. So to do this, we just put some insecticides in a bottle, and this bottle, we put it in a water bath. And this water bath allowed us to control for temperature, because mosquito nets are put in places that, I mean, there is a range of temperature. Most of the laboratory experiments we do to test nets and so on, they are standard conditions at 25 degrees. But we know that increasing temperature, we will increase volatility. So we try to mimic two of the temperatures that we can find in these places where people use the mosquito nets. And this is a very simple dispositif, which we have air that is, we push air with a pump. We filter this air so it's a clean air with active charcoal. And then this air goes inside the bottle where there is the insecticide, which is going to be a little bit more or less volatilized. And this air will be pushed through a tube towards a mosquito that is standing on the net. So here, what we want to see whether if the mosquito sense something, it will have to show us that it's sensing something by a takeoff response. So basically, we move away. So that is fundamental proof whether these mosquitoes can detect something. And we had the risk that they wouldn't do anything. So we cannot say whether they have detected or not, because we need a positive response. But luckily for us, we did this with insecticide sensitive and insecticide resistant strains that we have in the lab, reference strains. And we were very lucky because we found that there was a positive response. There was a little bit difference between the temperatures used. So When you do it at 35 degrees, there are more molecules, so the sensitivity is lower, so mosquitoes can detect it before. And but there was no difference between the strains, and we can say that mosquitoes were exhibiting takeoff responses when getting the insecticide on their bodies. Okay. So we confirmed that the takeoff response would be a very nice method to test for this sensory detection. But we wanted to know, yes, so we did this with reference strains in the lab, but more important is to ask the mosquitoes that are in the field. So he went to Burkina Faso, collected some larvae from field sites, and then asked the question to the mosquitoes with the same exactly the same experiment. And then we confirmed that the 3 species that he found, because first you need to do the experiments because they are very look-alike, they are very difficult to distinguish them morphologically. So you do first the experiment, and then you do a PCR to assess the species. And then what we saw is that regardless of the species, these are the 3 most common species transmitted malaria in Burkina Faso: Anopheles gambia, Anopheles colutia, and Anopheles arabiensis. All of them show this takeoff response, indicating that, hey, they sense insecticides without touching them. Okay. Later on, we ask ourselves, okay, so now that we know that they detect it, we want to know where they detect it. Which are the chemosensory organs involved in this insecticide detection? And just a very crash course of how it works. So mosquitoes and insects in general, they have different sensory organs, mainly localized in the head, which are the antenna, the palps, and also the just last part of the stylet where they bite. But also you can find them legs and on the wings, which are very badly known. We don't know what they do, these ones. Okay, these organs, they have all different receptors. And these receptors can be olfactory receptors, which are present in the stylet, or gustatory receptors and ionotropic receptors, which are found differently. They are expressed differently in the different organs. Okay, so mainly in the stylet, we find the gustatory and olfactory receptors. In the palps, we found olfactory, gustatory, and hygroreceptors, which are another kind. In the antennas, it's mainly, well, you will see, it's mainly olfactory receptors, but there are also thermoreceptors and hygroreceptors, and sometimes, gustatory receptors. But legs and wings, we find gustatory receptors. Okay, so how does it work a little bit? So if you see the antenna, like I will put that as an example, the antenna. If we zoom it, you will find like there are like particular structures that we call sensilla. For instance, this is one, this is another one, this is another one. So from this sensilla, inside the sensilla, we find olfactory sensory neurons in this case, but it can be chemosensory neurons. So where the odorants, the molecules, the chemicals that are present in the environment will go through the cuticle, be transported towards the membrane of these olfactory sensory neurons, and then on these membranes, we find the receptors, the olfactory, the gustatory, and the different kinds. And these ones, they are ligand-gated ion channels. So when the chemical gets into the receptor will open and will start the neural impulse. And this neural impulse will go to the brain of the insect, which then will decide what to do with this information. Okay, once we know this, we exactly did the same experiments, but we started eliminating the different organs of the mosquito. So first, we cut the antennas. Basically, it's a rapid ablation. It's a very rapid method to eliminate a sensory organ. So what we see here is that they were not responding to our controls, which is good. The controls were the solvent, where we developed the insecticide in this case. But with or without antenna, we could observe the behavioral response. So we thought like, okay, antennas are not playing a role in this detection, because if you ablate the organ that detects the insecticide, they shouldn't care, they shouldn't smell it, they shouldn't taste it, they shouldn't detected each other. Okay, so nothing happened there. I was very surprised not to find anything there. But then we went to the pulps. Here we reduced a little bit, but it was not enough as to say that yeah, there was something going on. So pulps are not involved in the insecticide detection, not the proboscis either. So we thought like, okay, what is left? Then we asked, what about feet? But here for our, no, that is, yeah, no. So here, I hope I didn't mess. Yeah, okay. Okay, good. Okay, so now we ask, okay, if there is nothing on the head, what about if we cut everything? And because, I mean, you can see here, there was a little bit of response, a little bit, and here it was lower. So we thought perhaps it's a combination and they compensate if one is not there or whatever. So here we try to cut all the organs. But first, when you cut all the organs, you need to make sure that they're still exhibiting the behavior that you're looking for, this take-off response. Then, for that, we use as a control DEET, which is a repellent molecule that we know that acts through the legs. So they could exhibit, even though they were not very well. I mean, you could see them that they were a bit depressed, not having inputs from the world. And but we didn't find any difference here, so we thought, okay, our system works. So we'll try to continue and ask them. So here we saw that the take-off response was reduced, but it was not completely abolished. So we thought, okay, there is something going on, somewhere else. And then we went to the legs. And for the legs, our behavioral experiment doesn't allow us to cut the legs because otherwise we wouldn't have seen anything. Yeah, so we put resin. We used resin, epoxy resin, which used with UV. So you kind of like paint them. You put socks, the mosquitoes, and then we tested again with these socks because they look like a bit, they were working funnily. We used a repellent that we know that works through the antennas. And in this case, we confirmed that they could still take off. We validated our experiment, and then we asked about the permethrin, and here we found that it was completely abolished. So we couldn't see any difference between here. So we thought, okay, now we know that insecticides volatilize in some sense that are present at the legs. And if you cover them, the mosquito doesn't send them anymore. So we're very happy to found this. But we went further. We wanted to ask electrophysiologically whether we could see these responses. And for those of you that don't really know what electrophysiology is, these are techniques that allow us to know which compounds are detected by insects in this case. So basically what we do, we put two electrodes, one on each side. One is the base electrode, which is usually in mosquitoes, we use the whole head. And the other one, we put it in the extremity of the organ we want to record. So in this case, you can see that there is antenna inserted here. And then once we have there, we have the activity of the neural population, which is present in this sensory organ. And then through here, we puff different stimuli. And then if we see deflections, it means that the insect detects this molecule. If you don't see any deflection, it means that it's nothing for the mosquito. So we did this with the Darcy, and we found that it's a dose response. So you need kind of like a high concentration of permethrin to start getting responses, but you really get these responses. If these are 95 confidence intervals, so if they don't touch the zero, means that it's significant. So for that, we also used a positive control, that compounds that we know that are detected by these organs, to see that everything was all right. And we use them before and after, because these are insecticides that are act in the neural system. So after stimulating with a lot of insecticides, this could compromise the responses that we see. That there was a little bit of reduction, but still they could sense. And here we see that there is a response. So we were happy to prove this as well electrophysiologically. We went to the pulpy, and here there was absolutely no detection whatsoever. And then when we went to the antenna, we saw that they could detect insecticides through the antenna. And that was, for me, really, really, really nice to discover, because it means that they can sense them, and it's much more sensitive than the tarsi. It can sense this molecule through the antennas, but it doesn't mean anything for them. So they need to detect it through the tarsi to exhibit a behavioral response. So the information of the same molecule, depending on the organ that is detecting, it means completely different from for the mosquito. Okay. And we went further because of this result of the antenna. We thought, like, okay, we should go and try to find some other receptors that can detect these insecticides. So, profiting of the work of a colleague in Sweden, Ulrika Regnell, that he is there organizing all the receptors, olfactory receptors of the Anopheles mosquito, and using the Drosophila melanogaster heterologous expression. So meaning that you have Drosophila melanogaster, which in one of the olfactory sensory neurons, it is empty. So you can put the olfactory receptor of no matter which insect is there, and then you can go and do very fine electrophysiology. So this electrophysiology, the other one that I show you, it is called an electroantennogram. Basically, you record from the whole population of neurons. But here in this electrophysiology, you go to just one sensilla. So you will record on the neurons which are in this sensilla, usually are two, and one of them is empty, and you can put the OR that you want. In this case, we had a couple of candidates. One didn't work in this expression in Drosophila melanogaster, but the other one really worked. It's called Anopheles gambiae, or olfactory receptor 30, and we could show that it really responds to pyrethroids in general, because we have permethrin, deltamethrin, alpha-cypermethrin, most of the insecticides that we've been using, but also to degradation compounds and other plant defensive compounds. Okay, so we were very happy to see the results that at least one olfactory receptor is responding to these compounds. Then, now we are working on the tarsi. How about the time? It's okay?
I will let you know.
Okay, so now we are working on the tarsi with my collaborators in Argentina, which are specialized in these gustatory receptors, to see if we can find any receptors there that can detect these insecticides. Okay, so take-home message. Anopheles gambiae, insecticide sensitive and resistant, these reference strains, detect pyrethroids without contact. That is clear. But also feel collecting mosquitoes of different species. So it's kind of like a generalized kind of like a response or way of detection. And I wonder whether other insects can sense the same thing, which would be excellent, and I expect them to do. Then escape responses are mainly elicited by the information coming from the tarsi. But there are at least two sensory organs involved in the detection, tarsi and the antenna. And the question of why is this redundant? My hypothesis here is what I was saying to you before about this could help a lot for the learning process. Meaning, imagine that you're a mosquito, you smell insecticides, you are very sensitive to this, you start detecting the molecule, but for you, it doesn't mean anything. Okay, but once you touch it, once you get close enough, or the concentration is high enough that you detect it through the tarsi, you will get a behavioral, usually it's an aversive response. Okay, next time you smell this, you could associate that the first time you didn't feel very well, that you didn't, because even though if you are resistant, they kind of like, they don't really like insecticides. They have this irritant effect that you don't die, but you don't get well. So they could use this as a way of learning associatively these compounds. On which for me it's more important, and now we are starting to go deeper into this, is that the same as I was telling before, the same molecule, the information that the molecule carries is different depending on what you from where you detect it. Imagine yourselves like cinnamon. Cinnamon for us, I mean, for most of us, you smell it and it's very nice. If you put a spoon of cinnamon in your mouth, it's completely horrible. I mean, the information is different, and you might or might not like it. And then pyrethroids, we can say that are agonists of at least one olfactory receptor in mosquitoes. Perhaps there are others. And it's highly likely that there are other receptors, like for instance in the tarsi, as if you remember, there are not olfactory receptors in tarsi. There are olfactory receptors, but also these ones, TRPs, which are very conserved receptors, which are they belong to a family of nociceptors. So meaning these are receptors that express or give information about pain, or about cold and hot. And they are very conserved. All the animals and the insects, they have them very well expressed in the tarsi, not only, but. Okay, so next step is to go to the field and ask whether this sensory detection could promote this avoidance plastically, basically. They smell it and then they will stay outside and wait for the host to go out and get bitten or bitten, or well, you understand. But also, and I have a PhD student and a master student that just started now, Sylvain and Clément Jacques. And Sylvain, his thesis is about learning in mosquitoes. And he has been doing wonderful experiments to show that mosquitoes, well, we knew already in other species, in Aedes species, but also in Anopheles species, we can confirm that they can associatively learn chemical compounds if you put the good reward. So now he has been working with repellents, with the questions like, could mosquitoes learn from repellents? If imagine that they are in a room, there is a repellent, but finally, because the repellents are not at the concentration we use them, they are not extremely toxic for mosquitoes, but they smell bad. That is just a feeling, it's just a sensation. But if they get bitten, if they bite, they might associate this appetitively, because they had a reward. They had a blood meal. There was this information in the environment with repellents. And then next time they will use these repellents to say, like, okay, there was a human, last time it was okay, I will bite. And it seems to work very well, but it's a matter of concentration. So relax. So low concentrations, mosquitoes can learn until at a certain threshold of this of concentration, the repellent effect takes over and they don't learn anymore. Okay, which is very nice. And I think, well, that is speculation, but I think it's related to the different organs that are detecting at high concentrations. Perhaps are other organs and other receptors that are detecting these molecules and saying, hey, no way. Okay, and the Clément is going to do this with insecticides, which is part of the Silver's thesis as well, and whether they can learn Anopheles mosquitoes appetitive insecticides or aversively, which could help, could increase the behavioral resistance that we see in the field. And I will thank all the people that was involved in this, like saying to all these people involved in this project and others, and especially the funding agencies, the INR, the IRD, Paul Valéry, Muse, Risk and Vectors, when it was, and the CNRS for giving me money for studying this course. Thank you very much.
Thank you, David. We have plenty of time for questions. Yes, Alex.
Yes, thank you, David. It's pretty amazing. I was wondering, you don't find any differences between the insecticide resistant one and the sensitive one? Wouldn't you have expected some difference where the sensitive one would benefit from escaping faster once it detects it? So what does that mean that there is no difference?
It's what we thought. That's why we tried both kind of like strains. Our hypothesis was exactly this one, that or perhaps the opposite, resistance wouldn't care because but they do. And because of the same thing, I think there is some information from the insecticides, perhaps coming from the evolution point of view. The molecules, the insecticide molecules come from plants, and they are defensive compounds, so they don't like it. So basically, it's something in them that if this molecule is present, they just don't go.
So resistance to persisting, you would have imagined that the sensitive one would have learned to.
Yes.
But like it's those who you've been too slow, you've been dead. Or...
It can be also the mode of action of these insecticides. These insecticides act on voltage-gated channels. So which are regulating the firing of the neurons. And here we are talking about receptors, sensory receptors. So perhaps it's the amount, like when we are volatilizing the amount of insecticide that we like the mosquito gets, is not enough as to get into these voltage channels and have a much wider response. But sensory or chemoreceptors are much more sensitive in this case. But I mean, I was surprised by that.
If you change the dose, or
sorry?
If you change the dosage, if it's like a big dose, maybe you could see what difference.
Yes, much bigger than we used to. It's just like kind of even dangerous, I think.
I remember where there have been hypersensitive strain described the resistance mechanism. So they detect at very, very low doses and they escape.
Yes.
Have you used this kind of strain?
No, not in mosquitoes. I was not aware in mosquitoes. There's all the studies in Drosophila with DDT that they could select this for hypersensitive strains, and they would see that the time the insect spent in contact with insecticides was much reduced. And then they were surviving because the amount of insecticide getting to this. But I haven't heard about hypersensitive mosquito strains to insecticides. I'm sure there are.
Thank you for the presentation. Following up on the learning process, I mean, I didn't get whether in your experiment, when you did the experiment on mosquito, had the chance to learn, or would it be possible to take in larvae from the lab and then grow them up without pesticides, so they have no opportunity to learn anything? And would then expel?
I didn't go into the details with the experimental protocol with this associative learning. We are using adults. So basically, you have an adult that will fit in the presence of a given chemical. In this case, it was different repellents, and now we are going to start with insecticides. So just the fact of fitting, getting a reward, it can be what is it, like sugar water, because mosquitoes eat nectar, sugar water, but the females, they need the blood meal to lay their eggs. So we've been using sugar water as a reward, also blood as a reward, and they can associate. So basically, this is a Pavlovian protocol, very standardized with a lot of controls, and it's a huge amount of work that Silver is achieving right now. So basically, you give them the opportunity to fit 3 times, and then you just present the molecule, and then you observe what they do. And as the dog of Pavlov, they will start biting just for the presence of this molecule that they shouldn't. I mean, in the beginning, we knew or we test whether this molecule gives kind of like a behavioral effect, and you don't see anything. But once associated, it is. And this is appetitive, and then it can be also aversive. But what you say is experience, whether the presence during the larval development could learn something. It's a very good question, which we haven't.
But then, and so the learning process is about rewards, but then the first answer to just take off or not, because you detect, has nothing to do with learning or not learning.
No. Okay. No, this is kind of like a reflex response.
I think it's related because I was first of all, I have to say I was impressed by how you cut all these organs and you put socks
Yes.
on the mosquitoes. Your PhD was especially awarded for his patience. I was still, like I had a hard time. Now your answer helped me a little bit better with the learning, because I was also thinking like that maybe just they don't care. Somehow you need to show that if you have this, you are visiting more in your case, you go more often into the house if that repellent is present. And I was just thinking, well, it's maybe you don't care. That's why you keep on doing it. Why you're not learning anymore. It's not about learning. It's about that you think it bothers them, but it doesn't bother them so much. And I couldn't see the learning now with the reward. I could see.
Yes.
So it helps, but still, how could you do that really?
So
leave them up. Like you can feel that they don't care so much. It's just not lethal enough. They just keep on going.
Yes, sure. I mean, mosquitoes will always try to bite. The thing is that you can speed up this decision making by learning, like having information from the past. You can go faster. You can go.
But if the dose is bigger,
learn
to die. So
yes,
there's no.
That's the thing. That's why we are testing this, because there are other cognitive processes that can be going on. For instance, Silver has been showing that kind of like there could be habituation processes, which are learning processes non-associative in a way that is non-associative. For instance, if you expose mosquitoes to a high concentration of insecticides, they won't care anymore. Sorry, not insecticides, repellents. They won't care anymore later on. So they won't be repelled by these insecticides. And he has showed that it's not a matter of our sensory kind of like, how should I say, like the receptors don't work anymore. It's just a cognitive process that they eliminated this sound, this habituation process. For instance, you are in a room, no one can hear the ventilation sound anymore, because you get your brain just like, but if you pay attention, you can hear it. So the same thing goes with the repellents. Like after a while, you don't pay attention anymore. And as I was saying, the concentration we use, they are not extremely toxic unless you're kind of drinking. It's just a sensory input that your brain can eliminate. So this is another kind of learning process, non-associative. Here we wanted to ask, like, can they learn? So if I put one repellent, the mosquito will come and bite at the same because of this, because the repellent will be much more volatile, they will be everywhere, and they can use this. And this could be appetitive or could be aversive. So you could associate a reward or appetitive or punishment. So you sense the smell, you get some kind of like punishment, a toxic effect, or something that it's written for you. Next time, since you are very sensitive with the tennis, for instance, to this smell, you won't go anymore. So you will reinforce the action of repellents. So that are the questions that Silver is trying to ask. And with repellents, but also with insecticides. And insecticides, they could be appetitive, but what I expect to be more kind of like, most probably what we're going to get is the aversive learning. So once you smell this, you get some shock, well, because you put yourself into a mosquito net or on the insecticide-treated wall. Next time you smell this, you won't go, because last time I got so you will reinforce this repellent effect. But since we are trying to kill mosquitoes with insecticides, it won't be very good for vector control, because the mosquito won't go. In houses, they won't go once they smell insecticides. And I wonder what this was the same with the reverse insects in agriculture. So they won't go, because last time they got it and they posted, they stand on a leaf where the insecticide was. Next time they got a sublethal dose, and next time they won't go. So they will wait, they will go to the neighbor, which is not true.
there any other questions in the room or online? No? Yeah, perhaps I have one. So I'm wondering, how do you interpret the taking off response? Behavioral. So you interpret that as an aversive behavioral response, or no?
No, it's just a response.
Just a response.
Yeah, because we haven't checked out. I mean, for instance, we could have used another chemical compound that we know that is not toxic or it's even appetitive and we could have, because it's out of context, we could have seen this takeoff response. For instance, this is a very nice example. Mosquitoes love heat, especially temperatures around 35, 37 degrees, because we are hosts, we are at this temperature, and they will use this. But whenever they decide to go, if you approach 37 degrees to the mosquitoes, they will go away. And because it's out of context, it's all the background. So here, what we measure is a takeoff response, which is a proxy of a detection, something, sensory detection, something.
So the follow-up question, coming back to the behavioral exploitation. So you expect the resistant, genotypically, physiologically resistant mosquitoes to somehow in the field can become either through learning attracted toward washed bed nets, impregnated bed nets. That's what you showed.
What they showed is that what they found, that washing bed nets, specific bed nets, you would find more mosquitoes in experimental huts when these nets were present.
So now they become attractive to the mosquitoes.
To the mosquito net.
Yeah, the mosquito net becomes attractive.
Yeah.
And then how experimentally could you demonstrate that the taking off response of genotypically resistant mosquitoes is an attractive behavioral response rather than aversive?
I didn't get it. So how would I test it?
Yeah, how can you demonstrate that your response, the taking off, can be attractive? I was thinking about the food deprivation and how food deprivation can actually influence your behavioral response.
Yes. Well, it's a matter of motivation status.
Yeah.
So if you food deprive or water deprive an insect, they will be more eager to go to this. So they will be perhaps less sensitive to certain molecules. Then, because they are more motivated, they just, otherwise I'll die. So I need to feed. So yeah, it's a matter of trade-off. But I didn't get the thing of the takeoff response and the food deprivation. So they will be more motivated. They will go, but they'll find ways. It's a matter of, it's always decision making. If you are very hungry or you need water, you will try to do as good as you can, unless there is something innate that tells you don't go there, otherwise you'll die. So insecticides can be this because of this irritant effect they have and so on. So it's a matter of deciding at this moment. Yeah, the second.
Do you start them in your experiments, the mosquitoes or not?
For not on the insecticides, all the experiments for motivating them for the associative learning, yes, because if they are well fed, they won't go. Okay. And you can spend like 3 days before they go and eat and they associate this. So we motivate them to go and eat. Yeah.
Thank you. Other questions? Yeah. No? All right. So thank you very much, David.